Hi, I'm Saad, and I'm co-founder and CEO of Panluminate. Today, I'd like to introduce you to a revolutionary new platform technology we call Panoptics. This is a technology which grew out of my PhD thesis here at Yale, and which we think will pave the way to true precision medicine. The fundamental premise of precision medicine is pretty simple. Take a sample from a patient, analyze it to identify critical biomarkers, and then develop a course of treatment that meets the precise needs of that individual. That, at least, is the dream. The reality, however, is a bit more complicated. It's been 20 years since the development of next-generation sequencing, and this dream still remains very distant. And so while everyone is still iterating on incremental improvements, we think the solution calls for something radically different, a solution that goes back to basics, back to the fundamentals of science. So at Panluminate, we have reimagined the entire process, and we innovated physically, chemically, informatically, and optically. And then we brought those innovations back together into a single integrated platform we call Panoptics. Panoptics is revolutionary because it's the only tool set that offers a complete solution to spatial biology. It is what we call progressive subcellular interrogation. This is kidney tissue. We start by physically expanding it slightly and then labeling it to identify cell neighborhoods. After that, this allows us to look more closely at individual cells at single cell resolution. However, we do this in 3 dimensions, unlike most single cell spatial omics companies. But what is truly revolutionary and why we're very different is that we're the only tool where you can take the same piece of tissue and then go subcellular, and even down to the protein level at the resolution of electron microscopy, which has never been achieved before on a light microscope. And this is because we expand the tissue even more and we label it again, merging molecular and structural information to reveal cellular mechanics and function, which is the essence of biomarkers. 2 proprietary technologies enable Panoptics. The first is our ability to radically expand tissue samples 10,000 times in volume. And the second is game-changing labeling chemistry that allows us to do unlimited multiplexing in 3 dimensions and label not only proteins, but DNA, RNA, lipids, etc., without doing any damage to the structure. I focused on the kidney because it's our first priority market, but our KOLs have told us that there's a real need for this technology in brain pathologies and in cancer research, with the lead indication in breast cancer. So let me explain where we are as a company. When we met a year ago, we were still mostly in academic collaboration mode. Today, we are a fully commercial operation. We have over 20 paying customers. We have customers from pharmaceutical companies. We have customers from startups and from universities that are outside the Yale bubble. In terms of technology, last year we had 1 foundational patent based mostly on my thesis. Today, we have this revolutionary, groundbreaking multiplex technology that I talked about. And we also improved our core processing protocols internally, and we think that our processing experience itself represents a great competitive advantage. Operationally, last year I was on my own. I had great advisors, but it was mostly just myself. Today, we have 5 full-time employees, including new senior managers to oversee platform development and applications and optimizations. We formed a scientific advisory board, which includes specialists to advise us on polymer chemistry and labeling technologies. In short, we're a very different organization today. So where do we go from here? This is our development pathway. Essentially, in stage 1, we think about the stage as being data creation. So we're optimizing our protocols and learning from customers. Then we want to move straight into automation, creating a box to automate sample processing and imaging. And in the final stage, we actually want to monetize the data, make use of it, turn it into clinical and diagnostic tools to be used by people outside of our laboratory. So how do we take that first step? We are currently raising seed funding. with a goal to raise $5 million to support us through 2025. And we would love to talk to any of you who may be interested in actually realizing this dream of precision medicine. And with that, thank you.